Welcome to ANA Investigates, a monthly podcast featuring the latest in neurology and neuroscience, brought to you by the American Neurological Association. I'm your host, Adeline Goss. Today, we'll introduce you to the next generation of ANA scientists. You may remember a new event at this year's ANA meeting called the Emerging Scholars Sessions, where we heard presentations from our junior ANA members about their cutting-edge research. Those talks were judged based on scientific merit and presentation quality, and today we'll introduce you to the four winners. This year's Emerging Scholars are Dr. Gus Davis, Dr. Juliane Gust, Dr. Jenn Orthmann-Murphy, and Dr. Xilma Ortiz-Gonzalez. They were interviewed by Dr. Michelle Johansen, a vascular neurologist and assistant professor of neurology at Johns Hopkins. Here's Michelle.
Welcome, everyone, to a special podcast featuring the winners of the 2020 ANA Emerging Scholars Lecture Series. I'm hoping that we'll have opportunity for the audience really to learn about who you are and for you to give us some insight into how you got to where you are in your research career. So, Dr. Davis, I would love to start with you, and then we'll just go through the group. So might you be able to introduce yourself?
Sure, I'd be happy to. And thank you so much to the ANA and all the committees for this recognition. I'm originally from Georgia. I did my undergraduate work at Emory University in Atlanta, and then started medical school and completed my PhD in neuroscience also at Emory before following my wife to Vanderbilt University to complete my medical degree. Then I moved to St. Louis to do my neurology residency at Washington University, and then remained at WashU for fellowship training in movement disorders and postdoctoral scientific training with David Holtzman. I'm currently an assistant professor of neurology at WashU, and I started my own laboratory about 2 years ago.
That's wonderful. Well, thank you for being here. We're going to be excited to hear about your research here in a minute. Dr. Gust, might you be able to introduce yourself?
Hello, I'm Juliane Gust. I am originally from Germany, actually, as you can tell by my accent. So I moved to Seattle before undergrad from Germany. I did all my medical training in Seattle. I'm a child neurologist at Seattle Children's now. And I work on the neurotoxicity of cancer immunotherapy. And I've been leading my own small lab for the last 2.5 years.
Well, thank you for being here as well. And then next we have Dr. Orthmann-Murphy.
Hello, thanks for having us. I am an assistant professor of neurology at the University of Pennsylvania, where I actually did most of my training, including undergrad, MSTP, neurology residency, and then I did do a postdoctoral and clinical fellowship in neuroimmunology at Hopkins before coming back here about 2 years ago, when I started a clinic taking care of people with MS, but also this undiagnosed white matter disorders neurogenetics clinic, which was the subject of my talk. And I have a lab looking at the basic science of remyelination and myelin disorders.
That's fabulous. And then finally, last but certainly not least, we have Dr. Ortiz-Gonzalez.
Thank you again, everybody. I think this is a great opportunity and it's an honor to be here. My name is Xilma Ortiz-Gonzalez. I was born in a small town in Puerto Rico where I did my undergrad at University of Puerto Rico in Cayey. And then I didn't know any better about snow or cold, so I just moved straight to Minnesota. And that's where I did my MD, PhD. I did my PhD in neuroscience. And after that, I came to Philadelphia, to Children's Hospital of Philadelphia, to do a residency in child neurology, where I stayed to do a fellowship in neurogenetics. And currently, I am an assistant professor in the Division of Neurology, and my clinical practice is in the Pediatrics Neurogenetics and Epilepsy Neurogenetics Clinic. What I'm really interested in are pediatric neurodegenerative disorders, and that's what my talk in the ANA was about.
Thank you guys for taking the time to introduce yourselves. And, we took a few minutes in that podcast to do that because I think it's really important for people to recognize that there are multiple paths to become a clinician scientist, right? And you all are from different backgrounds. You all study different disease processes, and you all went about your career trajectories differently. And I think sometimes for early career scientists, when they're thinking about research efforts, particularly in the era of COVID, unfortunately, it can be difficult to say, this is such a long road, I don't know if I'm ever going to make it. And we're really grateful to be able to highlight each and every one of you. So I'm going to ask you to just summarize your research efforts for us in a few sentences, and then maybe the most exciting takeaways from your presentation, or where do you think your research in particular is going in the future? So Gus, let's go ahead and start with you.
Sure. So this is Gus Davis. My lab focuses on the molecular basis of protein aggregation and neurodegeneration in Parkinson's disease and related conditions, including dementia with Lewy bodies. We're particularly interested in apolipoprotein E, or ApoE, which is the strongest genetic risk factor for the development of dementia and Parkinson's disease, and influences the pathological aggregation of the alpha-synuclein protein. And so in the lab, we use a combination of approaches all the way from in vitro protein chemistry, all the way up through mouse models of protein aggregation and neurodegeneration in order to investigate novel molecular and cell biological mechanisms that underlie disease risk and progression. And I think the most exciting take-home message from my presentation is that ApoE genotype directly regulates pathological alpha-synuclein aggregation. So this genetic association of apolipoprotein E with Parkinson's disease dementia and dementia with Lewy bodies was previously thought perhaps to be mediated through the well-established role of ApoE in modulating amyloid pathology, which is one of the hallmarks of Alzheimer's disease and is frequently found as a co-pathology in Parkinson's disease, dementia, and dementia with Lewy bodies. But our finding suggests that ApoE also directly regulates alpha-synuclein pathology. And so that suggests that targeting ApoE may be a therapeutic avenue that could be used for synucleinopathies in addition to other neurodegenerative diseases.
So it's interesting that genetics seems to be a theme in many of your presentations. And so we'll go ahead and go to Dr. Gust, Eula, to tell us a little bit about your topic. And then where do you think that your research is headed? Because I heard you during your presentation mention several different routes that you thought your findings could go in the future.
Yeah, thank you. So my research actually started very serendipitously by being in the right place at the right time. Seattle is a hotbed of development in cancer immunotherapy, and I was a resident in 2014 when we started to get neurology consults for patients receiving CAR T cells, which are the patient's own T cells that are reprogrammed to recognize the cancer cells and destroy the cancer. And about 40% of the patients who receive CAR T cells develop a neurologic syndrome, which can be relatively mild, just confusion and some language problems, but in rare cases leads to actually fatal cerebral edema. So this was completely unexplained, and I was right in the middle of it. And so I started kind of a two-track program of one, finding a model that we can use to mechanistically explain this and find ways to intervene and prevent it, and then also building infrastructure for collecting patient data to really have a very strong clinical phenotyping and be able to do correlative studies such as cytokine analysis to better understand what the mechanisms are of neurotoxicity. What I'm doing now is I, in the lab, study a mouse model of CAR T cell neurotoxicity. And then I'm also busy with being part of international and national consortia to study the long-term outcomes in CAR T cell neurotoxicity.
So on the theme of genetics, Dr. Orthmann-Murphy, I know that you had mentioned in your introduction you're fascinated with undiagnosed white matter disorders. So please tell us a little bit about your research and where you think, if someone were listening to your research, perhaps the most exciting takeaway from your presentation.
Thank you. I basically was trained as a myelin biologist starting in grad school, where I was studying how if you screw up the gap junction channels that form between astrocytes and oligodendrocytes can result in a devastating dysmyelinating disease. So even though I was doing basic science, it was based on a genetic disorder. And as I went through training and wanted to learn how to take care of people with MS, and as a postdoctoral fellow, just sort of getting frustrated with the fact that there was a small cohort of people that we just never were able to identify a diagnosis, I was in the right place at the right time, or I was just lucky to be surrounded by mentors who were like, sure, go figure this out. And then I reached out to people who totally let me go hang out with them and ask them a million questions and figure out how you do a genetic workup on someone who's an adult presenting, which was not something that I had learned as a resident. So that when I decided to come to Penn, where they already had an adult neurogenetics clinic and the ability to work with genetic counselors to evaluate patients, I could start to have referrals come to me that had white matter disorders that didn't fit these more typical diagnoses and start to do clinical genetic testing. And after about 2 years of doing this, I think the biggest takeaway of what I presented was that basically we found a diagnosis in more than a quarter of these patients. So it's worth doing this workup in adult patients to find a genetic disorder. Because on the one hand, if they're getting misdiagnosed with MS, they might be getting disease-modifying therapies that aren't doing anything and could be causing harm. And on the other hand, if we actually know what the diagnosis is, but we don't have a treatment, we now have the seed to go study and figure out how to treat these diseases. And so I collaborate with Adeline Vanderver, who started this myelin disorders biorepository project, which is a natural history study that's longitudinal, where we collect blood samples and basically everything that we can think of to understand how these disorders progress. And with patient samples, we could start to figure out what actually is happening in cells that are expressing this mutation, and one day even test potential interventions.
I think that's an excellent summary. And I think that in neurology research in particular, people from outside the neurology field can be frustrated when there are disease processes that we do not understand, particularly when we have nothing to offer patients after they've been identified. So your research will certainly be high yield in the future. Thank you for telling us about it. Dr. Ortiz-Gonzalez, please, we would be thrilled to hear a little bit more about iron metabolism and some of the genetic identification that you were able to do in your research. So please summarize your efforts for us, and then what do you think is the most exciting takeaway from your presentation?
Thanks again. So I come from the pediatric world again, where we have been doing more genetic testing, I think a little bit more aggressively than in the adult world. But we still have the challenge where we are able to sometimes identify genetic etiology, but in the case of very rare disorders, sometimes you're like, so what do I do with that? You have a mutation in this gene, nobody's ever heard of this gene. How do I even prove that this is disease causing? So that's one of the things that I work on, and the work that I presented at the meeting was precisely with the FTH1 gene, which is a ferritin heavy chain encoding gene. How do we even know if this is what's causing the phenotype? So functional validation of novel genes is something that in the pediatric neurogenetics world, we're sort of pushing the envelope, trying to, especially as genetic techniques get better and we find more and more variants, we all have lots of variants and all of changes in our DNA, which one is the needle in the haystack? And which one is the one that's driving the phenotype? So in the case of the neurodegeneration with brain iron accumulation, or the MBIA syndromes, we're working on first showing that this gene causes the disease. But what I think was most exciting about this particular project is that we were able to not only say, yes, we feel like this is disease causing and this is why, but we also were thinking about the next step, which is, the hardest thing that we do in clinic is like, what do we do about it? And are there any potential interventions or treatments, and how do we come about those? And in this case, we tested the antisense oligonucleotide approach to try to suppress the mutant allele because it's a heterozygous dominant acting mutation. And the idea was that if it is a dominant negative effect, so there's still one good copy of ferritin and you have one that has a mutation that is altering the iron metabolism, then you potentially rescue it with an antisense oligo approach. So I think that's the sort of theme that I'm interested in, my lab is interested in, is not only what can we learn from these rare disorders in children, and so how do we better diagnose them, how do we do functional validation, but also can we move towards therapies.
Wonderful. I appreciate that summary. I think that's outstanding. I want to transition now globally to talk to all 4 of you as winners. I would love to hear from each of you about advice that you would give to those who are listening to our podcast today, who are either junior or early career investigators and are once again overwhelmed by the situation in which we find ourselves in this year of 2020, trying to search for funding, trying to keep labs open, trying to consent patients, and thinking, how in the world do I get myself established and continue this research? So Dr. Davis, I would love to hear from you about any advice that you would give to those who are listening to us today.
Sure. This, this is a huge challenge. I agree. I mean, this type of field is very challenging to begin with, and then add on the additional layers of complexity with COVID. I think finding the right mentor and then the right team of mentors to help guide career development is even more important now than it probably ever has been. So I think that's my biggest piece of advice is pick the area that's the most interesting and then seek out the mentors that can help to organize a plan and provide feedback on discrete steps along the way.
I guess I would just add, when Gus mentioned the mentoring team, I found that personally to be very true, that not necessarily one person is going to be able to be proficient in helping you in every aspect of your research. So there may be somebody who's very good in the clinical side and teaching you, how to best take care of your patients, while there may be other people who are going to be good at reading your grants and critiquing them, so you have the best chance of success when you submit a proposal, for example. So finding the right mix of people for the skills that you need to learn is very helpful.
This is Yula. Couple of things if there's anything to add, it would be don't be scared. Even if it seems daunting, it seems like maybe you can't do it and everybody else knows more than you do. There's being worried and being scared of failure is not going to be beneficial. I've had my share of stomach ulcers, and I will have more stomach ulcers trying to keep the funding secured, worrying about that my mouse model is completely wrong, that I'm barking up the wrong tree. And if you let yourself be, paralyzed by that, you're never going to get anywhere. The other thing I remind myself of is this German saying that says everybody just cooks with water, which means, nobody is doing magic. Everybody has to follow the same types of rules. So if you learn how the game is played, you can play along. So using your mentors to really help you understand how to get into these funding structures, how to get what you need to do what you need to do, and how to ask for that is invaluable.
This is Jen. I think that everyone here has described really well. I don't have really a lot to add, except for something that I found very intimidating early on. I was told that you have to love this to do your job. And I was like, I have no idea if I love this. It's hard and I don't know what's happening. It was very much how I started. But it turns out that over time, there are aspects of this that I do love. And that's like the nugget of what makes me want to come in every day and do an experiment or review data now with students. It's that excitement that there's potential and hope for something. And, whatever it is, it doesn't matter. That is your motivator that gives you that excitement to, finally sit down and write that paragraph that you need to do, then go for it. Or realize that I don't know. And who do you have to ask to answer that I don't know? Is there an expert that you can go to help you? Or do you need to become that person?
Well, that was outstanding advice. Thank you for adding that. I think that being open and realizing that you cannot do this alone, and being willing to approach someone, and also realizing that there are going to be days that you do not enjoy what you're doing, and that is okay. So thank you for adding that, Jen. We appreciate it. Well, we have come to the end of our discussion. I want to thank each and every one of you for being a part of this. This was outstanding. Once again, this was Dr. Davis, Dr. Gust, Dr. Orthmann-Murphy, and Dr. Ortiz-Gonzalez, who we would like to congratulate as our 2020 ANA Emerging Scholars Lecture Series winners. Your research is outstanding. Your career trajectory is different and yet inspirational for the people who are listening. And I'm sure that they're going to appreciate greatly the advice that you've given today. So thank you all so much.
Thank you, Michelle.
That was Dr. Michelle Johansen, Assistant Professor of Neurology at Johns Hopkins, interviewing the 4 ANA 2020 Emerging Scholars. For more information on the Emerging Scholars program, including recordings of the presentations from the ANA's annual meeting, please visit myana.org. That's it for our show today. You can hear more of ANA Investigates and submit your ideas for future episodes on our website. For ANA Investigates, I'm Adeline Goss.
This has been an episode of ANA Investigates, the official podcast of the American Neurological Association. Thanks for joining us for this episode. If you're not already a member of the ANA, please visit myana.org to learn more about the benefits available to ANA members, including access to an array of educational programs and resources, like online CME courses, webinars, the ANA's journals, the Annals of Neurology and the Annals of Clinical and Translational Neurology, and more. Dr. Adeline Goss is our host. Our producers are Drs. Romer Giucaden, Adeline Goss, Michelle Johansen, Clifton Gooch, Danny Bega, Megan Ritchie, and Jim Sigler. This is your editor, Jen Hurley. Copyright, the American Neurological Association.